Hi there, my name is Ho Chen Yao. I'm a nephrologist from Singapore General Hospital and I'm currently pursuing a PhD with the Translational Research Immunology Group at the University of Oxford. My research focus is on the use of cell therapy to reduce immunosuppression in kidney transplant recipients.
Hi QI, my name's Helen. I also work in the TRIG group and I'm an academic clinical lecturer training in plastic surgery with an interest in reconstructive transplantation.
Hi Helen, I suppose we are here to introduce the meeting report on the 6th International Sam Strober workshop on clinical immune tolerance that was published in the April issue of Transplantation. As many of you know, transplantation is a life-saving treatment for organ failure, but most, if not all, solid organ transplant recipients need to be on lifelong immunosuppression to prevent rejection. As a nephrologist, I've seen so many of my patients suffer from the bad side effects from immunosuppression. Therefore, researchers have been looking for ways to achieve tolerance, which is operationally the reduction or potentially even elimination of the use of immunosuppression without rejection. And cell therapy has emerged as one of the promising strategies. The leaders in this field gather every 2 years to discuss the latest advances and future directions. This report that Helen and I have helped to write summarizes the data and issues that were discussed during the meeting. So to me, I was feeling really privileged to be part of this meeting because it really felt like an all-star game for the field of clinical tolerance.
Yeah, it was really cool. So one of the first areas that was discussed were chimerism trials. So this is a strategy to achieve transplant tolerance which targets central tolerance with a combination of conditioning protocols, such as total lymphoid irradiation, which removes the patient's own immune cells before replacing them with donor immune cells, and this gives rise to hematopoietic chimerism. This can be transient or persistent, mixed where recipient and donor immune cells coexist, or full where all of the cells are donor-derived. This paper includes details of several studies that are reporting good short to medium-term outcomes for patients who are maintaining good kidney allograft function without the need for maintenance immunosuppression. However, from my own perspective, this seems to be quite an invasive strategy which has the potential for significant adverse events such as graft versus host disease, viral infections and post-transplant lymphoproliferative disorders. In the select cases where patients already require a bone marrow transplant, I think it may be useful, but I think we're developing better alternative strategies for most patients who do not, such as regulatory T-cell therapies, which QI will tell you more about.
Yes, Helen. So the other approach, other than the chimerism-based ones that Helen has already discussed, is based on the use of regulatory T cells. So regulatory T cells are a subset of T cells that occur naturally within our immune system, and its main function is to suppress immune responses. Most of these strategies involve extracting regulatory T cells from the patient's blood, processing and expanding them to large numbers, and then reinfusing them to the patient at some point after transplantation. This strategy perhaps focuses more on peripheral tolerance, where it doesn't necessarily eliminate the immune cells that are reactive to the transplanted tissue, but mediate their immune responses and prevent tissue damage. For me, what is really exciting is previously these strategies seem like science fiction, but they've actually progressed to actual clinical trials. And from so far, from the currently available evidence that we have summarized in the report, it seems that these strategies are safe and potentially effective. Moreover, while many of these trials have small sample sizes, many of them had long-term follow-up of more than 10 years. And interestingly, there also seems to be a common immune signature where we can see the infiltration of immune cells into the allograft tissues for patients who have received these therapies, but without immune dysfunction or graft dysfunction, suggesting a tolerogenic function for these infiltrates. Importantly, compared to some of the strategies that Helen has discussed previously, we have not managed to withdraw all immunosuppression in patients who have received these Tregs, and these protocols still need the patients to be on some form of immunosuppression, most commonly tacrolimus monotherapies. So I think we still have some ways to go.
So, as we've heard from QI, one alternative strategy is targeting the peripheral immune response using Treg therapies to tip the balance towards tolerance. However, these products rely on a living donor, therefore they exclude those who have had a deceased donor from this therapy, and they're also pretty time-consuming to make. Therefore, teams are trying to engineer Tregs, which will specifically target HLA-A2. This is one of the most common HLA types, and this could then be used as an off-the-shelf therapy for patients receiving an HLA-A2 positive transplant. There are also groups looking to engineer Tregs to promote the stability of their suppressive phenotype and to produce Tregs resistant to tacrolimus, which is one of those immunosuppressants that QI mentioned earlier. So these advances are showing promise for delivering more personalised treatments to transplant recipients.
Unfortunately, Helen and I do not have enough time to go through the other details of the report, but in it, you will find a fairly comprehensive summary of the state-of-the-art for cell therapy research for transplant tolerance. The report summarizes the current clinical trial protocols and the latest results. It also discusses the use of alternative cell types such as dendritic cells, future directions such as the application of these therapies for autoimmune diseases, immune monitoring strategies, and the potential remaining challenges. Thank you for tuning in. We hope that you have some time to go through the report in some detail and enjoy reading.